So let's welcome Liang. He's from Stanford.
Okay, good. So I'd like to start today by thanking the organizers for the opportunity to speak. So the big question that I'm going to talk about today is, how does an adult organ maintain its size over the course of an animal's lifetime? So let's take a cartoon organ here where you have differentiated cells and stem cells. Over time, as individual differentiated cells carry out their function, they accrue damage or age, and eventually they are eliminated. At the same time, stem cells are constantly dividing to produce new progeny. So here, it's important to recognize that in order for an organ to stay the same size over time, the rates at which old cells are lost and new cells are produced must precisely equal each other. And so that's the question: how exactly is this balance achieved? To understand, to study this, we're using the Drosophila midgut as our model. And the midgut, as many previous speakers have already introduced, is a simple digestive organ that is constantly renewed through the activity of dividing stem cells. So let's look a little bit closer here. And what we see is that the gut is predominantly comprised of differentiated enterocytes, shown here in blue. And interspersed within these enterocytes are the stem cells in green. And these stem cells are the only dividing cells in the organ, and their divisions produce new enterocytes to replace the old ones. Now, for the midgut experts in the room, I want to point out briefly that our data is exclusive to the R4A and B compartments of the midgut. And so everything I'll be showing you today is limited to this region. So in my early experiments, I found that over the course of turnover, the Drosophila midgut is actually able to maintain a constant cell number. And this suggests that enterocyte loss and stem cell division must be happening at equal rates. But now the question becomes, well, how exactly is this relationship, what exactly is the nature of this relationship and what's going on here? And to address this, I propose a simple experiment. What happens if you perturb the rate of enterocyte loss by blocking apoptosis? What happens to the rate of stem cell division? Now, one possibility is that when you block enterocytes from dying, stem cells will still continue to divide at their normal fixed rate. And here, and this has actually been shown to be true for a number of developing organs, and here you would expect the organ to actually increase in size over time. The other possibility is that when you block enterocytes from dying, stem cells can somehow detect this, and they can slow down their divisions to compensate and try to keep the organ at the correct size. And in this way, you can think of stem cell division as being coupled to enterocyte loss. But to actually do the experiment, we need two things. The first thing we need is a way to block enterocytes from apoptosing. And here we can use the enterocyte-specific driver MEXGAL4 to express the caspase inhibitor P35. We also need an independent way of labeling stem cells and tracking their divisions. And here we're going to use a heat shock flip-based mechanism to do so and induce the clones that you see on the right-hand side. And by looking at the sizes of these individual stem cell clones, we can get an estimate of how many times single stem cells have divided. So what is the actual result? Normally, when you induce stem cell clones, what you see is that there is a distribution of different clone sizes, and this is exactly what you would expect. However, if you block enterocytes from apoptosing using P35, you actually see a significant decrease in the sizes of these stem cell clones, indicating that when enterocytes can't apoptose, stem cells divide less. So could this be a strategy that allows the organ to maintain the same size? Well, we quantify organ size by looking at the total number of cells in the organ in confocal three-dimensional reconstructions of the gut, and we see that indeed the organ size is preserved upon this perturbation, indicating that stem cell division is coupled to enterocyte apoptosis, and this provides a way to preserve organ size. The next question is, how is this happening on a mechanistic level? What are the molecular signals that tell stem cells to divide only when their neighboring cells are dying? And to make a long story short, I found that E-cadherin is a critical regulator of this process. So E-cadherin is a cell-cell adhesion protein, and in the Drosophila midgut, it's expressed by all of the gut cell types. Recall that when we block enterocytes from apoptosing, we actually see a significant reduction in the sizes of these stem cell clones. However, when we now additionally deplete E-cadherin from enterocytes, even though they still aren't dying, even though P35 is still being expressed, now stem cells fail to compensate and they divide as normal. So what is the overall organ-level consequence of this? Well, enterocytes can't apoptose, stem cells are now dividing again, and the organ actually becomes hyperplastic and it now fails to maintain the correct size. So E-cadherin seems to be clearly important in enterocytes in telling stem cells how much to divide. But what exactly is it doing? What mechanism is it going through? There's a number of possibilities. It could be interacting, or it could be dimerizing with stem cell E-cadherin. It could also be regulating a number of known proliferation pathways in the Drosophila midgut, including wingless, hippo, or EGFR. Now, when we investigated all of these possibilities, we found that it was actually this final case, the regulation of EGFR, that's important. So just to give you a brief primer about how EGFR is working in the Drosophila midgut, a number of labs, including Bruce Edgars and Rong Wen-Shi's, have found that EGFR is specifically expressed and activated in stem cells. And when this happens, the stem cell divides. So we can actually visualize this receptor activation by staining for phosphorylated ERK. And so these stem cells here that are ERK positive are presumably the ones that are going to eventually divide. However, when I overexpress E-cadherin in enterocytes, I find that there is a complete loss of EGFR activation in the tissue. And vice versa, when we deplete E-cadherin from enterocytes, now there is a hyperactivation and there's a significant increase in the number of ERK-positive stem cells. So you might then say, well, how do you know that this is actually specific to EGFR? How do you know ERK isn't labeling something else? Well, when we feed flies an EGFR-specific inhibitor, this hyperactivation effect entirely goes away. So enterocyte E-cadherin is repressing stem cell EGFR, but is this actually important for instructing division? And the answer seems to be yes. When we feed these flies an EGFR-specific inhibitor, we are able to rescue the excess divisions that's caused by loss of E-cadherin. And similarly, at the organ size level, the hyperplasia caused by loss of E-cadherin can also be rescued by inhibiting EGFR. So then the next question becomes, well, how exactly is E-cadherin on an enterocyte regulating EGFR on a stem cell? And here, I decided to do qPCR to get at this question. When we deplete E-cadherin, we don't actually see any effect on any of the EGF ligands, but what we do see is an induction of a gene called rhomboid. Now, rhomboid is a critical maturation factor in the pathway which produces EGF ligand, and it takes it from an intracellular inactive form into a secretable form that can then bind an EGF receptor on a stem cell. And as it turns out, rhomboid is a critical component of this signaling. When you deplete rhomboid, you're actually able to rescue the hyperactivation of EGFR that's caused by loss of E-cadherin. And so just to tell you, just to summarize what I've told you so far, normally in enterocytes, E-cadherin is repressing the expression of rhomboid, and in the absence of EGF ligand, stem cells don't divide. But then what exactly is happening when an enterocyte dies and you actually want stem cells to divide? Well, in data that I won't show you today, we find that dying enterocytes actually degrade E-cadherin, and this is consistent with many different studies that have been previously published. So if dying cells lose E-cadherin, could this allow for rhomboid to be induced, subsequently producing EGFs and activating stem cell division? Well, if this hypothesis were true, then you could make two predictions. The first thing you would predict is that you should see rhomboid get expressed in dying cells. The second thing you would predict is that the death of these apoptotic enterocytes should be necessary for stem cells to activate their EGFR. And so on the first point, when we look at rhomboid expression using Rho-Lag-Z, we see that amongst enterocytes, rhomboid is almost exclusively expressed in dying enterocytes like the ones I'm showing you here. Furthermore, we also see that the apoptosis of enterocytes seems to be necessary for the activation of EGFR. When we block apoptosis through P35, we see that there's a complete loss of receptor activation, and that this effect is also dependent upon both E-cadherin and rhomboid in the patterns you would expect. And so now finally, just to bring it home, looking back at organ size, the loss of, when we deplete rhomboid, we're actually able to rescue the hyperplasia that's caused by loss of E-cadherin. And so, taken all together, I'd like to propose the following model based on this data. When the organ is healthy and there are no dying enterocytes, E-cadherin is repressing rhomboid, and this limits the availability of ligand to activate stem cell division. However, when an enterocyte dies, the loss of cadherin in the dying enterocyte allows for rhomboid to be induced, which activates stem cell division and produces a new progeny to replace the old one, keeping the organ at the correct size. And so, ultimately, the take-home message is that dying enterocytes are driving the birth of their own replacements through activating stem cell division, and that this provides a strategy to maintain organ size over the course of an animal's lifetime. And with that, I'd like to finish by thanking my advisor, Lucy O'Brien, as well as the rest of my lab. This is actually drawn by another grad student in our lab. I'd also like to thank the Drosophila community for being a wonderful community to work with and for all the stocks and reagents and advice. And of course, my personal funding. I'd be happy to take any questions. Thank you.
Great talk. So previously it's been shown that Pseudomonas stress actually activates rhomboids in the midgut enterocytes. I was wondering if you think that the downregulation of E-cadherin may activate stress signaling, and have you looked at stress signaling?
Yeah, so that's a great question. I suspect that so we haven't looked at stress signaling in particular, but I suspect that when enterocytes die due to stress or due to injury, you would get very similar effects with downregulation of E-cadherin, which I predict would then subsequently upregulate rhomboid as well. Now that's something that we actually plan on looking at. It's on deck for experiments to do.
Do you know if E-cadherin's ability to repress rhomboid is dependent upon its ability to homophilically adhere to another molecule?
Could you repeat the last part of that question?
Does E-cadherin have to homophilically adhere to another E-cadherin molecule to repress rhomboid?
Right. What I can say is that when you overexpress a dominant negative version of E-cadherin, you seem to get very similar effects to overexpression of E-cadherin. And so I suspect that, so this is just speculation right now, and this is something we're trying to work out, but I suspect that this actually has more to do with the cytoplasmic tail of E-cadherin and not so much the homodimerization or engagement with other enterocytes or stem cells.
Great talk. Have you looked to see in any way if the stem cells most proximal to the dying enterocyte are the ones that are reactivated?
Yes, and I'm very glad you asked that question. So you can't obviously kill a cell and then look at it because the cell's gone. But what we can do is the next best thing, which is look at single enterocytes which you deplete E-cadherin from. And we can then ask, well, how far away are the stem cells affected? And at least in this case, what we see is, and this is consistent with the idea that EGF is being secreted, is that you see this sort of area of effect around the enterocyte that's affected up to about 40 microns or so. And so that's sort of the range of this signaling.
I have a naive question, but when you do an RNAi on rhomboid,
yeah.
Why doesn't the gut just disappear?
Does the gut just disappear?
Yeah.
So I have not. So what I haven't done is look at rhomboid RNAi alone, and that's actually something that I would love to do because, in fact, I think this really gets at the heart of it, right? Because if dying cells are still dying but they can't make rhomboid, then stem cells aren't getting the SOS signal to produce new enterocytes, and so then you might expect the gut to degenerate, and that would be my prediction, and that's actually something I plan on doing in the near future.
May I have a last question? I mean, E-cadherin is one of the best-known tumor suppressors in bowel cancer. Have you thought about looking into this in mammalian systems?
So I haven't thought about I haven't personally thought about looking at it in mammalian systems, but it does not escape my attention that both EGFR and E-cadherin are highly implicated in mammalian intestinal tumors. And so could something similar be happening where, because these genes are being mutated in mammals, you're actually getting tumor formation due to stem cell division now being at the wrong rate? I suspect that that's highly likely.
Yeah. Thanks, Yang.
Thank you.
So that concludes our session on stem cells. So please join me to thank the speakers for making this an awesome session.